See my screen okay?
Yeah. If you go to
Yeah.
screen share.
Yes. Good.
Yeah, perfect.
Thanks, Anne, and I'm very happy to update the community on behalf of myself, Giovanni Bernal, and all of our study teams, as well as all of the participants and everyone who's been involved in this very lengthy journey related to tamoxifen as a therapy for tubular myopathy. But as I start, I just want to recognize first the support of the My Tuber Trust and the Canadian Health Research Institute, who funded the preclinical work. And then for those who've ever been involved in an investigator-initiated non-industry sponsored trial, we recognize it takes a lot of contribution and a lot of support, and we are fortunate to have several groups who helped support the clinical trial, both in Canada and in the UK. And then the Sick Kids served as the sponsor for the trial, and very grateful for all of the support they provided to help this get going. So just to start, for those of you not familiar, just to answer the question why we even thought about tamoxifen as a potential therapeutic for tubular myopathy to begin with. And this was based on work that was performed in my laboratory as well as in the laboratory of Olivia Dorsey's. And we both independently discovered that tamoxifen is a very potent disease modifier in the mouse model of XLMTM. And this was shown, again, independently in two labs using multiple doses and dosing at multiple ages. And the upshot of the study was that at the highest dose and the optimal treatment, tamoxifen could more than triple survival of the MTM knockout mouse, and also improve critical domains such as animal strength, muscle force production, and resolve many of the pathologic changes seen in the muscle. So very promising based on the preclinical work as a potential therapy for patients with XLMTM, and also a drug with quite a bit of experience already in patients. Probably most of you are familiar with the use of tamoxifen as a chemotherapeutic for estrogen-responsive breast cancer. And so it was based on this that we considered doing a clinical trial evaluating tamoxifen. And this was the trial. It's called TAM4MTM, or tamoxifen for tubular myopathy. It's a randomized placebo-controlled crossover design study of tamoxifen for XLMTM. Our goal enrollment was between 12 and 15 patients, which was the optimized number to detect a significantly meaningful difference in a standard motor outcome measure called the MFM32. This is a standard measure that's been used in several muscle disease clinical trials to try to quantify features of muscle weakness and include elements like rolling, sitting, standing, and other movements as well. And it's been used previously in untreated XLMTM patients. Our inclusion criteria was quite broad. Starting range was, 6 months. Age range was 6 months to 60 years. In males with genetically confirmed XLMTM. There's a set of exclusion criteria, which I can review in more depth if people have questions, but basically anyone who was on another investigational product, who had a history of severe liver disease, or a history of thromboembolic events, or were on medications known to interact with tamoxifen, were excluded from the trial. So this was the study design. It was a phase 1/2 randomized placebo-controlled double-blinded trial. And so individuals received either tamoxifen at 10 milligram tablets or placebo. Again, the cohort was males aged greater than 6 months. The study duration was 48 weeks, and our goal enrollment was 12 to 15. Ultimately, we enrolled 7 individuals, but had to stop the trial prematurely due to adverse events, which I'll detail in a minute. So these were the study objectives. The primary objectives were to investigate the safety and tolerability of tamoxifen in males with XLMTM, and to determine the efficacy of tamoxifen based on motor function, and again, using the MFM32 as the primary outcome measure. These were some of the safety measures. They were standard blood work, including liver function studies, coagulation measures, and other blood cell counts, an ophthalmologic exam because there is a known association with ophthalmologic changes in tamoxifen, an ECG to measure cardiac and abdominal ultrasound, as well as a monthly clinical survey. And these were some of the secondary measures that were examined. We also looked at tamoxifen and tamoxifen metabolites and a novel biomarker that we're in the process of developing, and this analysis is still underway. So again, our study product was tamoxifen, and this formulation that we used was from a company called Apotex, and this is a generic version of tamoxifen. Happy to field questions about developing a repurposed drug. No non-generic pharmaceutical actually manufactures tamoxifen any longer, so the only access was through generic companies, and only Apotex was willing to give us tamoxifen. We had to pay for it, but they were the only ones to allow us to pay for it. And that created some of the restrictions we had in terms of being able to use this in the US. And we did twice daily dosing, so 10 milligrams twice a day. There was also a placebo that was manufactured by Apotex to look exactly like the tamoxifen, and this went to a biodistributor who then anonymously labeled the product as either as to all look the same, and it was either tamoxifen or placebo. And then participants took either tamoxifen or placebo for 6 months each with a washout period. So this is what the design looked like. There was a screening period where we did 2 measures independently, both 3 months before and at trial start. The initial 6-month phase, which was either drug or placebo, and this was randomized, a 3-month washout, and then a second phase of either drug or placebo. This was the frequency of assessments, just to give everyone a feel for what went into the trial and how often individuals who participate in the trial were monitored. And this is our study update. So again, our original target was between 12 to 15 individuals, and our original target sites were Sick Kids, Great Ormond Street, NIH, and Laurie Children's. For reasons related to the lack of an IND package at the FDA for this formulation of generic tamoxifen, we were never able to onboard US sites. And so we only had Sick Kids and Great Ormond Street participating. We enrolled 7 patients, all of whom successfully completed the study. And again, we're not able to onboard US patients due to logistical barriers. Unfortunately, the study was terminated due to serious adverse events in 2 study subjects. And I will detail those individuals in a minute. And we're still in the process of analyzing the effectiveness data from the 7 subjects that completed the trial. And we anticipate having results in the spring of 2025. So 2 individuals, as I mentioned, had serious adverse events. The first individual was coded as having hepatobiliary disorder and worsening liver function. This was noted 6 weeks after starting investigational product and was associated with elevated laboratory studies, including ALT, AST, which are 2 measures of liver function, and bile acids. The bilirubin, which is a measure of cholestasis, remained normal through the whole study. Based on these laboratory studies and consultation with first expert in liver disease, the drug study was stopped, and our DSMB, which is a Data Safety Monitoring Board, which had been organized prior to the study, this board was available for consultation to evaluate this adverse event. They recommended breaking the blind, and we discovered that the individual was taking tamoxifen. The recommendation was to stop the drug until the laboratory studies resolved. And over 3 months, all the laboratory studies resolved to normal, and the investigational project was restarted without difficulty, and the individual completed the study. I should note that the laboratory abnormalities never increased again, and that the individual never had any clinical signs or symptoms of hepatobiliary disease. It was only laboratory studies. And this was rated as possibly related to the study product. Then there was a second adverse event of acute cholestasis. This individual developed elevated bilirubins and bile salts in serum measures 4 weeks after starting the investigational product. The bilirubins continued to rise, as did now ALT and AST abnormalities. And at that point, the study drug was withheld, and the DSMB was again consulted. They recommended breaking the blind, and we noted that the patient was in fact on tamoxifen. This individual developed clinical symptoms of cholestasis, including itching, which is the generic term for pruritus is the clinical term for itching, and jaundice, which is a yellowing of the skin and also of the sclera of the eyes. I should note that the bilirubin peaked at close to 300, which is an extremely high level. Based on the laboratory abnormalities, the patient ended up having a prolonged hospitalization and having the liver dysfunction managed with medications, did not require any surgeries or interventional procedures. Eventually, all of the abnormalities resolved by approximately 2 months after their start. But based on this event and the previous SAE, it was the recommendation of the Data Safety Monitoring Board that we stopped the trial due to the potential association of tamoxifen with cholestatic liver disease in individuals with XLMTM. So to summarize, we conducted a randomized placebo-controlled crossover trial of tamoxifen for XLMTM. We successfully enrolled 7 individuals, all of whom completed the study. The study was terminated due to liver-related SAEs in 2 subjects in the study. At present, we're completing the data analysis and hope in the early 2025 to have a full presentation of all of the study data. We're also now going back and re-examining tamoxifen in our preclinical models to see if we can better understand the relationship between the drug and liver injury. And our recommendation based on this trial at this point is that we would encourage patients and families as well as clinicians to not use tamoxifen off-label for XLMTM until we have further data regarding its safety as well as its potential effectiveness. And just a quick note, this is not the first clinical trial for XLMTM where liver disease has been noted. I think probably everyone on the call is familiar with the Aspiro gene therapy clinical trial, the results of which were published in Lancet Neurology last year. This is a study of gene replacement therapy using AAV8, driving the expression of or delivering the gene MTM1. The AAV8 is delivered to the muscle and the liver, but the MTM1 is only expressed in the muscle. 24 patients have been treated in the clinical trial thus far. I think everyone is aware of the tragic occurrence of 4 deaths related to liver disease from the Aspiro trial. These individuals developed hepatobiliary cholestasis, similar to what we observed in the tamoxifen trial, but fortunately, tamoxifen trial, they were limited and reversible. This type of injury due to gene therapy has been unique for XLMTM and not seen in other AAV gene therapy trials, though liver injury in general is something that can be seen with AAV-mediated gene therapy. I should note that more information related to these cases are coming in the form of a publication with a manuscript that's currently in preparation. So just to conclude this part, the next steps related to liver and XLMTM, which you're going to hear about in just a minute. I should note that there's been an additional case report of 4 cases of cholestasis published a couple of years ago, and I think there's several of us who are aware of other cases of cholestasis that have been identified in patients with XLMTM, which leads to the challenge and goal that I think we're facing moving forward. One is to understand the natural history of liver disease in XLMTM. We need to understand which patients will get liver disease, why they get it, what those precipitants are, what the consequences are clinically, and how to potentially treat the liver disease when it develops. And these will be the subject of several studies that are being planned, which will be detailed in just a minute. We also need to understand the why. Why is it that the loss of MTM1 causes liver disease? And also, can we identify potential strategies to treat it? And this is the subject of lots of preclinical work that's happening in several laboratories, including my own. And I think we'll hear a bit about these if there's time permitting at the end of the webinar. So I think at this point, I'll pause. Could take questions on the tamoxifen part of things before we move on. Does that make sense, Anne?
Yeah, it does. And actually, there's a question here from Wendy. Can you see that in the chat?
Yes. So great question from Wendy. What are the implications for women who are female carriers of MTM1 mutation and who may be prescribed tamoxifen for breast cancer or other comorbidities? So I think just taking the caveat that the advice is based not on hard data that because there is not any at present. I would say two things. One, there has yet, as far as I'm aware, to be a report of a female with an MTM1 mutation having cholestatic liver disease. And I think to me, it's quite possible that only affected males will develop cholestatic liver disease. I can't say that for certain, but I think in adults, it's something that's relatively easily identified. And so I don't know that I wouldn't think that we're missing clinically significant liver disease in females with MTM1 mutation. And as such, I would therefore say my recommendation would be if tamoxifen is medically necessary for treatment of breast cancer, that there would be no contraindication to pursuing it. Probably would just make sense to alert the prescribing clinician of a potential association that at this point has not been shown. So I would say it would be safe to continue the current treatment strategy, but just to be aware and potentially incorporate that into the monitoring of safety of the drug. Ooh,
Jim, I've been sent a question offline. Can I ask you?
okay.
Um, in your announcement, you said that the liver test results returned to normal after stopping tamoxifen, and that you're certain there won't be long-term effects. At the same time, you state that you clearly advise against off-label use. Is there a reason why you don't simply say that in the case of off-label use, you should check the liver values closely, and if the values are elevated, discontinue the tamoxifen immediately?
So that's a good point. I think so if there's two questions incorporated there. One is whether there's any long-term concern of episodes of cholestasis. I think in general, our feeling is that having one episode may not cause any long-term problems. And certainly in the screening period that we've had for tamoxifen, we were looking to see, did this predispose to liver disease developing right away more chronically? And that's not what we saw. On the other hand, I think it's really hard to say that 25 years down the road, having episodes of cholestasis may not have an impact. So I can't say one way or the other. It's probably a good question for our hepatology experts who know similar conditions better. So that's one thing I'd say. I think the other issue I would point out is that these two individuals were examined in a clinical trial setting where there was very frequent monitoring and where the changes were flagged almost immediately after the onset of liver changes. And fortunately, they were both reversible, though I must say that the second individual was quite sick and was in the intensive care unit for several days. So, and reflecting on the Aspiro trial, where 4 individuals developed severe cholestasis that ultimately proved to be fatal, I don't know that we can say for sure that if you develop an episode of cholestasis related to tamoxifen, that it's always going to be reversible. And so I think to me, that's where I would have a pause. While it seems straightforward to say, let's try it. And if you develop liver changes just to stop, I think the concern is we don't really have great therapies for reversing it. I think in the 2 individuals that developed the changes in tamoxifen, it just was time and not necessarily any medical interventions per se. And so, I mean, I think you could envision a scenario where if you develop this on tamoxifen, even when you stop the medicine, if it was already severe enough that it could cause real lasting injury to the liver. And so I guess that's where my caution comes from.
Can I add a comment,
Yeah.
Jim? Because I agree absolutely with what you were saying, but also it's very difficult, to early identify, signs of cholestasis. Some of these laboratory abnormalities basically developed progressively, So if you don't have a very tight clinical monitoring, as you can only do in a clinical trial, it's very difficult to capture and be sure when this is reversible or not. So it's very, we should strongly recommend to do this under, a proper study and so on, as we did for this clinical trial.
And actually linked to that question in the Q&A, you'll see there's a question from Dan. Are the liver side effects of tamoxifen usage treatable while maintaining tamoxifen use?
Again, I think we don't have great therapeutic options yet for the cholestasis. I mean, there were some things that we've tried, and we had some hepatology experts who were had tried some medical management. We had some procedures that we could use if things had gotten worse. It's not clear whether those would have worked or not. So I think the answer is yes, there are some therapeutic options. There's probably a small number of pediatric hepatologists in the world who are familiar enough with this type of liver condition to really have good thoughts on how to treat it. So again, would definitely need someone who's quite expert in this type of cholestatic liver disease to do that. So yeah, I don't know if Dan, I answered the question sufficiently. I see a couple other questions. So to Allison's question, the biomarker is something that we have a manuscript in submission that's being reviewed related to that. So probably more on that soon. It's not tied to liver failure. It's actually something we developed as a muscle biomarker. So it's responsive to severity and change in the muscle, at least in the mouse model. The publication from the liver is again all of the similar authors that have been part of the Spiro clinical trial. I'm not sure the specifics of that as well. It's driven again by Astellas who as the sponsor. And to Aaron's question, so no, we haven't actually. We're still in the data analysis process because we were blinded to the efficacy measures or to the patients, and so don't yet have that data, Aaron. That's something when we do have it, and it's all I'll compile it. Happy to share with this as soon as we do.
And I've been sent another question, Jim, did all patients show the effects positive or negative, or was there a huge variety? And i.e., could it be that only a subgroup of patients would have liver problems, while for another subgroup, tamoxifen has only positive effects? But I guess
so again, yeah, we don't know anything about the effectiveness at this point. That's still data that we're analyzing, and we're just now putting together all of the data and aggregating it and doing the biostatistics. So it's not like I'm hiding anything. I honestly don't have an idea about effectiveness. The side effects, the cholestasis, the laboratory cholestasis was only identified in 2 of the 7 individuals. I think in general, one of the open questions that makes this all challenging to think about is that it's clearly only a subset of individuals that with any given precipitant develop cholestasis. And I don't think there's a good way to predict who that is at this point. So I think that makes it challenging to know who might be able to safely receive medicines that have a risk of promoting cholestasis. So sorry, I can't be more specific, but I think that's hopefully something we'll learn through the natural history study in terms of what the precipitants are and whether there are specific subgroups of XLMT patients who are more or less susceptible. Yes, that's getting to Sarah's question. So far, Sarah, no idea why it is that some patients have developed it and others haven't. No obvious, there's nothing necessarily obvious in the history. My hunch is that as we start looking, many individuals with XLMTM will have had silent episodes of cholestasis in their life. And so I think this is, again, I would stress the critical importance of things like the forthcoming Excel natural history study, as well as the MTM global registry and the liver questionnaire that's been developed, because these are what's going to help us try to answer those questions of why some individuals are developing cholestasis and others aren't, and what those potential precipitants are. Maybe it's as simple as every person that developed cholestasis had something that happened just before, but we won't be able to identify that until we start collecting this data more broadly.
Thanks, Jim. They're really thorough, thoughtful answers. Thank you. Oh, one more from Kate from MDUK.
We have to wait to analyze. So yeah, good question, Kate. I mean, nobody had clinically significant elevated bile salts or bilirubins otherwise outside of the two individuals, because that was an SAE criteria. So nobody else met the liver-related SAE criteria, at least by laboratory studies. Whether there's going to fall in a category of higher than normal, but not meeting that bar, I don't know yet. Because again, we haven't looked at all the data. Yeah, so just to give everyone the general, so this is something that's happened pretty recently, and what we've been doing since the second episode of occurred, and the DSMB recommended terminating the trial, is dealing with all of the necessary documentation that goes along with having to stop a trial prematurely. So with Health Canada, with FDA, with UK regulators, with clinicaltrials.gov, these were all. So we've had to take a pause on the data element of things to get these important regulatory documents completed, so that we're in compliance and everything was safe and dealt with in a way that's was needed.
That's great. Thank you, Jim. That's really, really helpful. Would you like to move on to your next slides now? Is that we seem to have come to...
Yeah, I was going to hand it over to Giovanni.
Yeah, I can share my screen if
I'll stop. Yep.
Yes, thank you. Can you see my screen now? Yeah.
Yep.
Let me put it full. So yeah. Good afternoon, everyone. I echo what was mentioning at the beginning, that we are grateful to all the patients who took part in this trial, to the families, the Myotubular Trust and the Gosh Charity who funded the study in the UK, because as you can easily imagine, it's not easy to do a repurposing drug clinical trial, because, there is very little financial gain from a study using an already existing medication. But every time, although this was somehow a study that we needed to stop, every time we learn something new. So I guess that, despite everything, we managed to move ahead a step, towards the identification of a potential treatment, understanding the disease, and so on. And this is why studies, natural history studies like this one, that is actually funded by Astellas, will try to look more in details about the hepatobiliary problems in patients with XL-MTM. And as you see, this is an observational study. The primary objective will be to identify hepatobiliary health, so the occurrence of these problems in the patients and how they evolve over 48 weeks of assessment. And then there will be additional objectives like identify if some genetic variants of MTM1 are more susceptible to develop the liver complications, if there is any possible association with environmental factors. We will see that data on diet will be collected and so on. And also what is the impact in terms of healthcare utilization. And these are all information that are critical when we will have a potential treatment, when we deal with the regulators in assessing, the cost benefit of drugs. As I said, this is a non-interventional project where there will be no investigational drug that will be administered, and everything will be performed somehow according to the local standard of care. There is an anticipation to involve at least 15 to 25 specialist sites across the UK, US, and Canada. And this will mainly focus on the pediatric population. So patients have to be male patients with MTM1 mutation and an age below the age of 18 years. They anticipate to recruit up to 50 patients. As I mentioned, 48 weeks of observation with 6 weekly appointments. And essentially, there will be liver function tests, the ones that also Jim mentioned in our protocol, looking specifically at some of the new liver function markers like bile acids or GGT or bilirubin, dietary habits, the liver ultrasound, and other lab samples. And again, this is a protocol that has been designed to limit as much as possible also the burden to the patients, so everything should be done according to what is already part of the routine care of the patients. And this was my last one. I will hand over back to you, Jim. Who was next?
Maybe we'll take if there's any questions about the Excel study, and then if not, I think we're going to hear about the registry.
Yeah. You can always come back with questions on the Excel study later on at the end anyway. Now I'd like to thank Julie Bohill, who is the curator of the myotubular and central nuclear myopathy patient registry, to particularly cover what the registry's been doing to collect valuable data on the liver. This is where we as the broad patient community can make our contribution to a greater understanding of the liver. Thank you, Julie.
Thanks very much, Anne. Can everybody hear me okay? Yeah. Okay. Okay, I'd like to firstly... Oh, excuse me. Just bear with me. Seems to have some...
Do you want me to try? Shall I try and share your presentation?
Yeah, there seems to have been some sound on it and I'm not quite sure why. Yes, Anne, if you wouldn't mind, that would be great. Thank you. Okay. Yes, firstly, I'd like to thank Anne for...
Sorry, Julie, am I in yours or am I in mine? It looked like I might be...
Should be in yours.
Sorry, 1 second. Try again. Right. There?
Yeah.
Right.
And then just... Okay, right. Thanks so much.
Through the future, I'd like to run through
just bear with me.
That are managed and curated here at Newcastle University.
Just bear with me 1 moment.
No worries. There's never a webinar without a glitch these days, is there? Does anyone know a good song? You want to unmute yourself, Julie?
Okay, we think we've found the problem and corrected it. I do apologise about that. Okay. Yes, so I'd like to very firstly thank Anne and Mel for inviting me along to talk about the myotubular and central nuclear myopathy register. So there's myself and Lucy Hickson who curate the registry. We are based here at the John Walton Muscular Dystrophy Research Centre in Newcastle. The registry was initially developed and set up by the Myotubular Trust, but then transferred to the care and responsibility of Newcastle University, who it's got a wealth of knowledge and experience of running patient registries. And currently we run 3 UK registries and 3 international registries. So very quickly, in terms of the patient registry, it's a global registry. It's disease specific and it's intended primarily as a research database. And then the 3 categories of patients that we accept are living and deceased patients with diagnosed and medically confirmed MTM-CNM, and also female carriers of X-linked MTM, whether that's asymptomatic or symptomatic. We've currently got 541 patients on our registry, and consent is via an online registration through our portal. And I'll go on to the registration and the questionnaires in a moment. One of the key factors on the registry is that we would like genetic or biopsy reports from patients. It's a very easy process to upload. Any patients have any difficulty doing that, they can contact Lucy or myself, and we will upload the genetic reports. But the genetic reports is the critical thing in terms of access to clinical trials or to be part of a research study. We also have doctors nominated on the registry. So a patient, once they've registered with us, is able to nominate their treating doctor, and we then contact them to invite them to join and also to provide some clinical data. One of the key factors is to ensure that this registry, which is covering this very, very rare disease, has up-to-date registry data that tracks the natural history and the progression of this disease. So rather than this be a one-time login, we ask patients and we remind patients to go into the registry every 6 months to update the data so that we can track a patient's progression and how the disease is impacting them. One of the issues about the registry being in Newcastle is that we have NHS, we're governed by an NHS ethics. We have an independent steering group, and we're very grateful for Jim, who's on the call, Jim and Anna Bujbella, who are our co-chairs. And it's an independent steering committee with very valuable participants who come from patient organisations and are clinical experts. Okay, so our broad headings of patient registry data come under these blue boxes, and the ones highlighted there, the liver questionnaire and brain neurological conditions, are new questionnaires that have recently, in the last year and a half, been added to the registry. Okay, next slide, please. Okay, so very quickly, how does a registry work? Could you just click the next one as well, please? So patients log in to a secure platform. And it can be a patient, it can be a family member or a carer. They enter their data and answer a number of questions. As I've just mentioned, they can also nominate their treating doctor, who is then invited to join. Once registration is complete, that's where the curator's job comes in, and we review and validate the data to ensure it is of a high quality and complete. So at that point, if there is data that's incomplete or where a genetic information or genetic confirmation hasn't been provided, we then go back and do a follow-up with the patient to ensure the data is of a good standard. And this data is extremely valuable. I think in Jim's presentation and Giovanni's, the data collection is the really key point here, that this data from a rare community is what is needed in order to enhance and support research and inform on standards of care, and also for clinical trial feasibility and recruitment. Next slide, Anne. Okay, so how did the liver questionnaire come about? Many of you might know this, but very briefly, for those who are not aware, this was developed by a group called the MTM-CNM Liver Collaborative, and that was really pushed through very strongly by Anne Lennox and Erin Ward, who realised, on top of all the clinical trial information that was coming back about the impact of liver and obviously the very tragic Aspira outcomes, that there was also underlying liver conditions within the community where patients and families were recording that there was very severe cases of liver health. So the liver health questionnaire was developed collectively and collaboratively with liver experts and clinicians from around the world. And this questionnaire went live into the registry in 2003. And when you log on, this is what registration looks like. You are then led through a number of questions. Liver function is one of those. So if we just go through the liver questions, and some of the questions that we're going to see now, Giovanni and Jim have mentioned these things before. So we collect information on cholestasis, whether the patient has been diagnosed or not. And one thing I would like to say here is if when a patient comes to complete this questionnaire, if the answer is no, you've not been diagnosed with anything, the capture of that information is very, very important. It isn't just a case of, oh, I don't need to fill this in because I haven't been diagnosed. The collection of the no, I haven't is as valid as yes, I have been diagnosed, because we want to understand of the full population of the patients that we have on this registry, how many have and have not been diagnosed. So we ask, have you been diagnosed with a liver condition? As in those questions about liver condition at the point of being newborn, and then also liver symptoms. So we have patients on our registry who haven't been diagnosed with a liver condition, but they have liver symptoms. And I think Giovanni was mentioning that before. How do we know, what is the scale of liver issues within the community? So this is very important data. We also ask information about the lab tests, and this was covered earlier, about whether there's been increased levels of ALT or AST or increased bilirubin levels. We're also interested to know, has there been any hospitalizations as a result of abnormal liver tests, and whether these were planned hospitalizations or not. And also if there's been any liver imaging done, whether you've had a CT or an MRI or an ultrasound. Other questions that are asked in the liver questionnaire is, who reviews the liver health? How often how often are the checks undertaken, and are patients on any medications? And we also want to know when the medication started and ended, and if there's any liver, sorry, can we just go back one? If there've been any liver biopsies. And also the final question is, are there any other questions that perhaps the patient or family member would like to share with us, but the questions haven't enabled you to be able to provide that. So there's some free text there. So this questionnaire, as I mentioned earlier, went live in the registry in April 2023. I need to move on, but very quickly, we have now 187 patients have completed it. We've published, we've presented at 3 conferences, and this information is available on our website. And we are looking to do a publication in 2025 on the work of the Liver Collaborative and how they developed this questionnaire, and we will then be analysing the results. So it's a very brief whistle stop, but I'm very happy to take any questions now or after this webinar. Okay, thank you.
Thank you very much, Julie. That's really helpful. As you can see, the instructions of what you need to do if you've joined or haven't joined already are on here. Were there any immediate questions to that section? No. But Julie is always available for any further questions if people have any. I am now, one sec, going to share with you. Share. So now I would like to cover off the what else is being done in this field? When these liver challenges started, it's not that everybody paused and there was nothing happening for a while people tried to process this. Pretty much immediately, a number of different liver research projects have kicked off. As Giovanni outlined, Astellas are going to run a natural history study, but then at the Myotubular Trust, we just want to share with you that we have also been pretty much immediately finding excellent world-class research projects that we can use the funds raised for us by you, the patient community, to try to understand the liver even more. And we are very lucky that each of the project leaders of these projects have found the time to join us today to give you a brief overview of each of these Myotubular Trust funded projects that are to look into further into the liver. Anna, can I ask you to share your slides first? Do I still have you, Anna?
Yes.
Are you okay? Are you able to see me? Can't. Yeah.
Yes.
No.
Okay. Thank you. Okay. Thank you very much, and thanks for the invitation to participate in this call. I just have a couple of slides just to show that we are working on this aspect of myotubular myopathy. Let me see if I can share my screen just briefly. Okay. Okay, can you see?
Yeah.
Okay. Okay. So first, of course, I would like to thank Myotubular Trust for the funding of for our projects in general, and from early days on gene therapy, and more specifically on this last project. So, just as a brief reminder, this project that is funded by Myotubular Trust is to develop a new gene therapy vector for myotubular myopathy, taking into account new technological advances in the field. These are the various points that we are studying. So first, as I mentioned, just another AAV vector carrying the MTM1 coding sequence. Then as well, we are planning to study the feasibility of AAV vector readministration in the context of AAV8 seropositivity. As you know, I mean, once the AAV 8 vector is administrated, then there are antibodies that are developed, and then we are studying the possibility of re-administrating AAV vectors in that context. And we are as well interested in understanding better the liver pathology in XLMTM animal models under basal conditions, and as well after AAV transduction, and to generate an MTM1 deficient mouse model specifically in the liver. So all these projects are ongoing, so I cannot really share data with you, but just in the context of let me see, of the liver, just to say that we are characterizing the liver of animals that are deficient for myotubularine using different techniques, molecular techniques, biochemical and histological techniques. We've seen some changes, in particular at the transcriptomic level, that we are trying to understand and to validate. Some changes as well at histological level. And we are generating these liver-specific MTM1 deficient mouse model in order to see whether we can see a defect in the liver, especially in the long term, and as well under specific conditions like, for sure, AAV gene administration. So at this stage, this is what I can share. So the work is ongoing. We have some preliminary data, but that need to be confirmed.
Thank you very much, Anna, and thank you for your constant dedication to X-linked myotubular myopathy. You have utterly changed the field, and we are all incredibly grateful to you for the work you've done. And thank you for making time for us today as well. In order in which the grants were given, Jim, are you available to describe the grant that we have made to you in partnership with our wonderful friends at the UK Muscle Disease Charity, Muscular Dystrophy UK?
Yes,
Thank
yeah. So just quickly, so the overarching question related to the grant that was generously funded by My Cheaper Trust and MDUK relates to understanding the role of liver pathomechanisms, the loss of MTM1, and also the interplay with AAV and how potentially one could think about developing therapies to mitigate the liver injury associated with AAV. So just to remind everyone where things started, which is after the recent clinical trials, the very unexpected emergence of cholestatic liver disease, and this recognition that possibly or likely MTM is also a liver disease in addition to muscle disease, and the lack of animal models that have really shown us this. So we went back to our original zebrafish model where we astutely had figured out that there actually was a liver change in 2016, but then promptly ignored that fact. But now have gone back and looked again. And also we've been trying to address the question of why it is that the mouse model does not develop evidence of liver disease. So we've now gone on to show that the zebrafish model, so this is a zebrafish that lacks the MTM1 gene, develops a reproducible cholestatic-like liver disease. And so this will be a really excellent model for starting to study the whys of how this happens and what loss of MTM1, why it's causing the consequences in the liver. But the focus of the grant is really on, and this was published last year in JCI, so in case you're interested, the focus has been on the mouse model. And we asked the simple question, well, maybe it's, had the simple hypothesis that maybe it's that the environment in which we grow up our mice is responsible for protecting the livers with the loss of MTM1. And if we made some simple environmental changes, would that now bring out liver disease in the mice? And so our first step was by changing the diet of the mice to one that is less liver protective and more resembling human diets. And once we did that, we very quickly realized that the MTM mice develop a very obvious and reproducible liver disease that looks like what we're seeing in patients just with this simple diet change. And that's what's shown here. And this is now the springboard for all of the studies that we're going to be doing related to studying liver pathomechanisms, the loss of MTM. We also have an eye on potential therapy interventions. This is just one thing we've been looking at, which is the idea of delivering MTM1 exclusively to the liver using lipid nanoparticles as a way to protect it from environmental induced liver injury and hopefully also protect it from AAV induced liver injury. And finally, I should say that, and I think you're going to hear about this from the next presenter, we've also been working on cell models of the liver. So we've developed induced pluripotent stem cells with mutations in MTM1 and differentiated them into liver cells and then into liver organoids. And we see basically the same features that we see in the mouse liver, in the zebrafish liver, and also from what we understand has been described so far in the human livers with loss of MTM1. So these are going to be a really excellent model for studying as well. So I will stop with that.
Thank you very much, Jim. And Dr. Leonello and Professor Tedesco, I would really like to hear from you now too. And this is a grant that we are working in partnership with Great Ormond Street to provide to this team.
Thank you, and I will let Valentina to give a short presentation on that.
Thanks, Valentina.
Hello, can you see my screen?
Yes.
Perfect. Do you see the presentation?
Yes.
Okay, hello everybody. So I'm Valentina, I'm a postdoc in the laboratory of Professor Saverio Tedesco. So we work in UCL and the First District Institute in London. And just we want to give you a small overview of what we are doing in the lab and what we are doing for this project that started in May 2024 and will finish in 2027. So the main aim of the lab is actually to identify a model that is specific for each patient and that in a short time help us to better understand the disease mechanism. So for the reason, we started to develop a in vitro model of skeletal muscle. So to get closer to how the skeletal muscle works in the lab of in the Tedesco lab, we are culturing the cells in a 3D environment and not in the standard tissue culture environment, let's say in 2D environment. And these help us to better understand the progression of disease and to better understand how actually muscle behave. So the first aim of our project is indeed to model skeletal muscle derived from patient cells. And we differentiate this muscle in a 3D environment. And here you can see that actually the muscle that we generate in the lab from patient cell line, they are functionally active. So when we supplement, for example, electrical stimuli. In parallel, while I was developing this part of the project, I was extremely lucky because another postdoc in the team started to work on liver and for another project that we had ongoing in the lab. And actually we could get his, let's say, protocol and his ability to differentiate this patient cell line to liver cells and to generate liver spheroid. So the next step of the project that will start soon is actually also to generate the MTM1 liver spheroid, as you can see here in the slide. And this will help us to better understand actually how MTM1 patients can develop the disease that we heard like in this last hour. Finally, the last part of the project will be to merge these two tools, so the 3D muscle and the liver spheroid together in order to test different therapies. So we mainly will focus mainly on gene therapy. So we will start with AAVs. We already know that our muscle can be easily transduced with AAV. And we will verify if actually we can have an effect on DV on the liver spheroid that we generate from patients cell line. So this actually, as I told you, this grant started in May 2024. And he will finish in 2027. So we thank a lot the Metubular Trust and the GOSH to help us for this part of the grant. And we also work we are part of a consortium. Saverio is actually my group leader. He's is the coordinator of a big consortium that is generating different AV serotype, specifically for skeletal muscle. So we will have access also to all the different type of AV that is currently generating to test in this, let's say in this platform, multi-organelle platform. And this is all the team, all our team. So we are really happy to be part on board with the Metubular Trust. So thank you. I'm happy to get questions if needed.
Thank you very much, Valentina. And we are very happy to be working with you too. If anybody has a burning question now, I'm conscious of people's time. If anyone has a burning question now, you can pop it into the Q&A. Alternatively, the email address that you use to register, contact@Myotubular Trust, you could follow up with any questions to us and we can direct them to whoever here today might be the appropriate person to answer them. Sorry, excuse me. It's not been an easy time for the myotubular myopathy community. It's been it's been beyond heartbreaking when this liver situation began. And now, this is the final moment of closing down this particular road we were on. But while it is, as I say, beyond heartbreaking that this is what's happened in our community, what we do know we have, as well as an extraordinary dedicated community of patients and parents and who look after each other so well, but we also have this community of researchers, and you are exceptional and your commitment is outstanding. And without you, we would be in a hope-dashed situation where with you and your commitment, we can see there will be ways forward. We will find out what's going on with the liver. It's going to take all of us, patients and you guys, but we will find out. And we are very grateful to all of you, whether you're a researcher, somebody who has witnessed a trial not work out, or a patient and a parent who's logged onto the registry and made every effort to make sure we know what's going on in your family. We're grateful to you all. We're all in this together. And thank you also from Kate at MDUK, and thank you to MDUK for supporting us too. Thank you, everybody. Have a good evening. It's been lovely to be with you all. Thank you for your time.
Thank you.